Welcome to the WIONPodcast, where we explore fascinating stories and ideas from various fields. In this episode, we explore Decapeptide 12, a precision peptide gaining attention in pigmentation research for its potential to target the biological processes behind uneven skin tone. Researchers are now studying how this peptide works in the skin. The advent of peptide science has brought with it peptides with extremely targeted interactions with biological pathways. These include a Decapeptide 12, with a proposed function in pigmentation biology. It is a synthetic peptide which may be targeted to melanin synthesis mechanisms and is being evaluated as a research tool to better understand melanin synthesis molecular regulation. It has been speculated to have selective interactions with melanogenic pathways and has been used as a valuable subject in peptide engineering, molecular biology, and cellular signaling research. Peptides are short chains of amino acids and are basic signaling molecules in biological systems. They are smaller and easier to manipulate, so they might be able to be engineered to bind to a specific receptor, enzyme, or cellular pathway. This versatility has led to the increasing importance of synthetic peptides in the laboratory, where they are used to investigate biological processes and to develop new molecular models for scientific research. A synthetic peptide is one of these, known as Decapeptide 12. It consists of 10 amino acids that are designed to bind to the key regulators of melanogenesis. It is thought that its structure may allow it to interact selectively with enzymes that are associated with pigmentation, notably tyrosinase, which has a major role in the conversion of the amino acid tyrosine to melanin. Tyrosinase has been consistently found to be one of the important control points in pigmentation biology from scientific literature. The regulation of tyrosinase has been highlighted in reviews that appeared in physiological reviews, Biochimica et Biophysica Acta, and the International Journal of Molecular Sciences, in the field of understanding melanogenesis and pigment-related cellular mechanisms. In addition to tyrosinase, Decapeptide 12 is also being studied for its possible impacts on more global pathways in cellular communication that are related to pigmentation. Researchers are investigating the peptide's potential to influence regulatory proteins, such as microphthalmia-associated transcription factor, signaling pathways such as cyclic AMP, protein kinase A, and mitogen-activated protein kinases. These pathways, in turn, regulate the expression of pigmentation genes and cellular responses. Secondly, Decapeptide 12 is a rather small molecule, and therefore is a good peptide engineering model. The researchers are now able to make very specific changes in the sequences of amino acids and see how this affects the molecule's binding affinity, as well as its specific and biological functions. The modular approach will aid scientists in the design of next-generation peptides in more targeted biochemical research. Decapeptide 12 is proposed to be more specific than many naturally occurring peptides that act on several biological pathways. This type of molecular design is believed to facilitate targeted experimental investigations and is in line with a wider movement in peptide science towards precision research tools. Synthetic peptides are under continued research to gain a deeper understanding of the biology of pigmentation and its molecular regulation, and Decapeptide 12 remains significant. It is a promising case study for the use of engineered peptides to aid biochemical and cellular research due to its specific design and possible interaction with melanogenic pathways, and is still under investigation. The published studies on Decapeptide 12 in journals such as Physiological Reviews, Pigment Cell Research, Journal of Investigative Dermatology, Biochimica et Biophysica Acta, General Subjects, and the International Journal of Molecular Sciences have contributed to the scientific understanding of melanogenesis, tyrosinase regulation, and pigmentation signaling. Thank you for tuning in to this podcast. If you enjoyed this episode, make sure to like, share, and subscribe for more captivating tales from India and beyond.